The title of this article is Cognition Mediated Evolution of Low-Quality Floral Nectars. The authors of this article are Vladislav Nachev, Kai Petrostich, Clemens Winter, Alan Bond, Alan Camille, and Yorker Winter. In this slide, we are shown three different plants, two having the anatomy of each. This will lay ground for later on in the experiment when describing how the plants are pollinated. The general approach of this article was to describe and demonstrate how the evolution of plants is based on the consistency of pollination and its effects on the animals around it. The pictures on this slide of the ladybug, bird, and bat all demonstrate which animals and insects were used during this experiment. Other insects include bees. A determining factor in the evolution of pollinated versus diluted plants is the habitat and animal life surrounding it. Depending on how many other animals there are that are capable of moving pollen around, this will be an all-around factor. In order to conclude and study this, the researchers in this experiment synthesized this process by replicating it in the lab with living bats, hummingbirds, and other natural resources. The main question proposed by these researchers was if the pollinators really did have an effect on pollination and plant evolution. These graphs were taken directly from the experiment. As shown in figure B, the virtual plant population is shown by the results of bats and its pollination. This is taken between a 12-hour foraging session in order for the bats to locate the best given plants. This refers to the diluted and the most pollinated plants in the experiment. As shown in figures C and D, the results of this process are given. During the experiment, the researchers took data on how many independent decision makers, including bats and hummingbirds, pollinated the active or dormant flowers. After pollination, graphs show an estimate of how many plants became more active, if any. These results were recorded. As shown in the tables, the decision makers mainly pollinated the active pollinating plants more than the dormant or less active flowers. Their results showed that the flowers that were mainly active did increase in pollination. However, the less active plants also increased in pollination and became more active. In order to create these graphs, the number of plants, bats, and hummingbirds were all accounted for in the same number as their group for treatment and control groups. A series of trials were run over an extended period of time in order to replicate real-life scenarios. Prior to the experiment, these questions were thought of. The first question asked was, do animal life and their ability to move pollen around affect the pollination status of an individual plant? The reason of focus on bats and hummingbirds was due to their consistency of moving pollen from plant to area, therefore proposing the idea of using them for this experiment. The second question was, when pollinated, do the plants increase pollen or remain similar to the same without being pollinated? This question was based solely on the title. It explains how different flowers pollinated and less active react to being pollinated and if the long-term evolution coincides with these results. With the data gathered and questions answered, the researchers were able to test their hypothesis and finalize its validity. These researchers found that their predictions of the flowers being pollinated and becoming more active was indeed correct. They also found that the evolution rate of pollen depends very heavily on the decision makers, in this case the hummingbirds, bats, and insects. With this information, scientists will be able to determine pollination rates in different areas of the world, depending on animal life around the plants. This can help in floral advances and businesses, along with future scientific studies, if a number of pollinated or less active plants are needed.